A very good morning to all. In our last class, we have studied about the mitochondria and plastids. Now in this video, we are going to study about ribosomes. The ribosomes are also known as ribonucleoprotein. So these are the granular structures that are observed under electron microscope as dense particle and given name ribosomes by George Palade in 1953. So ribosomes are commonly known as protein factory as they help in the synthesis of proteins. The ribosomes are composed of ribonucleic acid, that is RNA, and protein, and are not surrounded by any membrane. So there is no membrane present outside the, or you can say this is not a membrane bounded organelle. The ribosomes are found in both prokaryotic cell as well as eukaryotic cells, but some exceptions are always there. So the exception is the mature sperm and mature mammalian RBC. In these cells, the ribosomes are absent. The ribosomes are of mainly two types according to the presence. So these two types are the cytoplasmic ribosomes and the ribosomes that are present in organelles. So the cytoplasmic ribosomes may be free ribosomes that can present in cytoplasm freely, or it is in the form of attached one that is attached to endoplasmic reticulum to form rough endoplasmic reticulum and nuclear membrane. And in organelles, the most important that one is mitochondria and plastids are also have their own ribosomes. The ribosomes are of two types according to their structure, that is ATS and 70S. So mainly ATS is present in eukaryote and prokaryotic cells, 70S is present. Eukaryote is also having 70S also. So here S stand for sedimentation coefficient or Swedberg constant that directly measures the density and the size of the ribosomes. Now, what is this units, or you can say the sedimentation units? These are the units that can settle down after the centrifugation process. सेंट्रीफ्यूगेशन प्रोसेस के बाद में जब ये यूनिट सेटल डाउन हो जाती है उसके बाद हम लोग इसको काउंट करते हैं, दैट इज द सेडिमेंटेशन यूनिट्स. So both 70S and 80S ribosomes are composed of two subunits, the larger and the smaller, as shown in the diagram. The larger unit is there and a smaller unit is there. So the largest is dome shaped. It is dome shaped and is always attached to membrane if it is attached ribosomes. अगर ये अटैच्ड है तो इसका लार्जर साइड हमेशा मेम्ब्रेन से, अगर हम ईआर की बात करें या न्यूक्लियर मेम्ब्रेन की बात करें तो जो हमारे वहां पे बाउंडेड राइबोसोम्स है उन बाउंडेड राइबोसोम्स में लार्जर यूनिट उनके साथ में अटैच होगी बाय द हेल्प ऑफ अ प्रोटीन दैट इज राइबोफोरिन प्रोटीन. Okay, a special protein that is riboforin protein that help in the attachment of the larger unit to the membranes. The smaller unit are always free. They get only attached to the larger one when the protein synthesis takes place. So the smaller unit in the form of granules wander here and there in cytoplasm and it get connect with the larger subunit when there is a protein synthesis process. So on the basis of combined settle down or sedimentation, the prokaryotic is having the 70S type of ribosomes, whereas eukaryote have 80S type of ribosomes. And if we are talking about their separate units, the prokaryote is having 30S, the smaller subunit, and the 50S, that is the larger unit, if they sediment separately. The molecular weight is 2.7106 Dalton, and the size of the prokaryotic ribosomes are about 20 nanometer, or you can call it 200 Armstrongs in diameter. The constituents of prokaryotic ribosomes are 35 to 40% of protein and 60 to 65% of RNA. As shown in the diagram, you will find out that in both the unit, the constituent particles, that are rRNA and proteins, are different. So there are three types of rRNA, or you can call it ribosomal RNA, in prokaryotic ribosomes, that is 16S, 23S and 5S. The 16S is present in smaller unit and the 23 and 5S is in the larger unit. And why these are three separates? Because of their number of nucleotides in it. If we are talking about protein, so the smaller unit is having 21 proteins and the larger subunit is having 34 protein in prokaryotes. Then if we are talking about eukaryotic cells, the eukaryotic cells is about 25 to 30 nanometer in diameter or 250 to 300 Armstrong in diameter, having 45% of RNA and 55% of protein. The subunits are 40S and 60S when it's sediment separately, but if it is sediment combined form, it is 80S. The molecular weight is 4 multiply 106 Dalton. And when we are talking about the types of ribosomal RNA, it is of 4 type here, that is 18S, 28S, 5.8S and 5S. Again, the smaller subunit is having 80S ribosomal RNA, whereas the larger one contains 28S, 5.8S and the 5S ribosomal RNA. So this is the structure of ribosomes that we have studied, that they are having two types of units, larger and smaller, having different composition of proteins and ribosomal RNA. Normally these are present separately in when it is in free form, but they combine together to form a complete ribosome structure when the process of protein synthesis takes place. So in this video, we have studied about the ribosomes structures.